Carbidopa and Parkinson's disease. How does it work? Carbidopa is a drug that blocks conversion of levodopa to dopamine outside of the central nervous system and thus inhibits unwanted side effects of levodopa on organs located outside of the central nervous system during management of Parkinson's disease. PD is associated with increased expression of inflammatory genes in peripheral and central nervous system, infiltration of immune cells into the brain, and increased numbers of activated memory/T cells. Animal models of PD have shown a critical role of T cells in inducing pathology in CNS. However, the effect of carbidopa on T cell responses in vivo is unknown. Carbidopa mitigated myelin oligonucleotide glycoprotein peptide fragment 35 to 55 induced experimental autoimmune encephalitis and collagen-induced arthritis in animal models. This suggests that in addition to blocking peripheral conversion of levodopa, carbidopa may inhibit T cell responses in PD individuals and implicate a potential therapeutic use of carbidopa in suppression of T cell mediated pathologies. Parkinson's disease is characterized by a loss of dopaminergic neurons in substantia nigra in the brain, resulting in decreased production of the neurotransmitter and messenger dopamine. Loss of dopamine is central to the development of PD. Levodopa, the dopamine precursor, is converted into dopamine by L-dopa decarboxylase. This leads to increased production of dopamine. Therefore, levodopa is very effective in the management of Parkinson's disease. DDC is expressed by neurons in the central nervous system, liver, kidney, pancreas, and T lymphocytes. Consumption of levodopa results in systematic production of dopamine in the central nervous system. In addition, activation of peripheral dopamine receptors results in nausea and vomiting. Carbidopa is an inhibitor of DDC and does not cross the blood-brain barrier, thus preferentially inhibiting the conversion of levodopa to dopamine outside of the brain. However, the effects of carbidopa on peripheral cells, specifically T lymphocytes, has not been studied in detail. The combination of levodopa and carbidopa is used to treat the symptoms of Parkinson's disease and Parkinson-like symptoms that may develop after encephalitis or injury to the central nervous system caused by carbon monoxide poisoning or manganese poisoning. Parkinson's symptoms include tremors, stiffness, and slowness of movement and are caused by a lack of dopamine, a natural substance we usually find in the brain. Levodopa is in a class of medications called central nervous system agents. It works by being converted to dopamine in the brain. Carbidopa is in a class of medications called decarboxylase inhibitors. It works by preventing the levodopa from being broken down before it reaches the brain. This allows for a lower dose of levodopa, which causes less nausea and vomiting. In the 1950s, anticholinergic agents were used in the treatment of PD. Anticholinergic are centrally acting antimuscarinic drugs such as trihexylphenidol, piperidine, phenidrin, and procyclidine. The molecular structure of carbidopa. X-ray crystallography is currently the most favored technique for structure determination of proteins and biological macromolecules. Increasingly, those interested in all branches of biological sciences require structural information to shed light on previously unanswered questions. Furthermore, the availability of a protein structure can provide a more detailed focus for future research. The extension of the technique to systems such as viruses, immune complexes, and protein nucleic acid complexes serves only to widen the appeal of crystallography. Structure-based drug design, site-directed mutagenesis, elucidation of enzyme mechanisms, and specificity of protein-ligand interactions are just a few of the areas in which X-ray crystallography has provided clarification. The aim of X-ray crystallography is to obtain a three-dimensional molecular structure from a crystal. A purified sample at high concentration is crystallized and the crystals are exposed to an X-ray beam. The resulting diffraction patterns can then be processed initially to yield information about the crystal packing symmetry and the size of the repeating unit that forms the crystal. This is obtained from the pattern of diffraction spots. The intensities of the spots can be used to determine the structure factors from which a map of the electron density can be calculated. Various methods can be used to improve the quality of this map until it is of sufficient clarity to permit the building of the molecular structure using the protein sequence. The resulting structure is then refined to fit the map more accurately and to adopt a thermodynamically favored conformation. This is the molecular structure of Carbidopa.